Imagine this for a second. A burglar is trying to pry open a window on the ground floor of, say, a huge 10-story building.
Right.
He hasn't even made it inside. I mean, he's barely scratched the paint with his crowbar. But instantly, and I mean instantly, no alarms, no guards running up the stairs. Every single window on the 10th floor slams shut and locks.
It's the perfect security system.
Yeah.
A total building-wide lockdown before the threat has even properly begun.
Exactly. And for the longest time, we had basically no idea how plans could pull this off. They don't have a nervous system.
Right. No brain to send out that burglar alert.
Yeah.
Yet somehow the signal gets from the bottom leaf to the top leaf in record time.
But today, we are not just guessing. We are going to be dissecting a huge breakthrough paper from Cell, published just this year, 2026, from Liu and his colleagues. They've finally figured out the machinery behind this magic trick.
And it has a name, systemic stomatal immunity, or SSIM for short.
And what's so cool is that this isn't just about plants fighting off bacteria. It's a genuine detective story. It's about, hidden codes in the genome we thought were just junk.
And that kind of cellular self-destruct mechanism that seals all the doors from the inside.
I do love a good self-destruct mechanism. So, our mission for this deep dive is to decode this paper pretty much sentence by sentence. We're going to follow this signal from the moment one leaf gets attacked to the moment the whole plant goes on red alert.
There's a lot to unpack. So to start, I think we have to talk about speed. Because speed is what makes SSIM so different from everything we thought we knew about plant immunity.
Right. We've talked about plant immunity before. And usually the conversation is about something called systemic acquired resistance, SAR. That's the textbook defense.
It is, yeah. SAR is the heavy artillery. It uses salicylic acid. It rewires the plant's genes. But it takes days to fully activate. This is slow burn.
Days?
Days. What Liu's team looked at here is completely different. They infected one local leaf, we'll call it leaf 6, with a common bacteria, Pseudomonas syringae.
Or they didn't even have to use the whole bacteria, right? They could just use PHLG22, which is basically a chemical piece of the bacteria's tail.
That's a great analogy. It's like showing the plant a mugshot of the bad guy without the bad guy actually being there.
Right.
So they show this mugshot to leaf 6. And then they just watch the distant leaves, leaf 7, leaf 5, leaf 8 leaves that haven't been touched at all. And within just 2.5 hours, the stomata on those distant leaves had slammed shut.
2.5 hours? That is ridiculously fast. That's not build an army speed, that's emergency broadcast speed.
Exactly.
And just to make sure everyone's with us, stomata are the little pores on a leaf's skin, right? They're for breathing.
Yeah. They let CO2 in for photosynthesis.
Yeah.
But they're also the front door for pathogens. If the stomata are open, bacteria just waltz right in.
Oh.
If you close them, the bacteria are stuck outside, they starve or they dry out.
So the plant feels a threat on one leaf and tells all the other leaves to lock their doors. But the big question, the one that's been bugging scientists for years, is what is the signal? What's the telegram that travels from leaf 6 to leaf 7 so fast?
This is where the real detective work begins. They knew it wasn't the usual suspect. It wasn't salicylic acid. They tested mutants that can't make it, and the system still worked.
So they were hunting for a ghost, a totally new molecule.
A new molecule. And to find it, they had to use a different tool. Usually to see what genes are active, scientists use a technique called RNA-seq. That tells you what's being copied from DNA to RNA.
Okay, but that's like looking at a library's checkout list. You know what books people took off the shelf, but you don't know if they're actually reading them?
That is the perfect way to put it. Just because the RNA transcript is there, that doesn't mean a protein is actually being made from it. So Liu's team used something called RiboSeq. It looks at the ribosomes themselves, the protein factories. So it tells you not what could be made, but what is actually being translated into a physical product right now.
The difference between a blueprint and the actual building, and what did they find?
Well, they found something really strange. They saw the ribosomes working away in a part of the genome that's, usually ignored. It's considered regulatory fluff or even just junk. They found a uORF.
A uORF. Okay, you're going to have to unpack that one for us.
Right. So uORF stands for Upstream Open Reading Frame. Picture a gene like a song on an album. The main song is the gene itself, in this case, a gene called MYB51.
Okay, MYB51.
And we know MYB51. It's involved in making defense chemicals. But before the main song starts, in that little bit of silence at the beginning of the track, there's this tiny hidden intro. That's the uORF. It sits in what's called the 5' untranslated region.
The intro track. So we've been skipping the intro for decades to get to the main song. But in this case, the intro was actually making something.
It was making the weapon. As soon as the plant was infected, this tiny little hidden region started churning out a small peptide, only 22 amino acids long.
Just a 22 amino acid message.
A tiny little telegram. And they gave it a name, USIC.
USIC.
That's a bit of a mouthful. uORF-encoded systemic stomatal immune conductor.
Okay, USIC is better. But what about the main gene, MYB51? Was it just a coincidence? Or is it involved?
That's the crazy part. They use gene editing to get rid of the main MYB51 gene, and the systemic signal, SSIM, still worked perfectly. But when they made a tiny mutation in just the uORF, just the intro track, the whole system broke.
Wow.
The local leaf could still fight back, but the distant leaves never got the memo. Their stomach just stayed wide open. The signal was gone.
So the secret message was in the hidden track all along. It really makes you wonder how much of what we call junk DNA is actually mission-critical code we just haven't learned how to read yet.
That is the billion-dollar question in genomics. So, okay, we have our signal molecule, this tiny peptide, can't has made in the infected leaf, but how does it get out?
Right. To ship a protein out of a cell, it usually needs a shipping label, a signal peptide. Does USIC have one?
Nope. Nothing. It's completely unlabeled cargo.
So how does it get past the cell membrane? How does it sneak out?
It uses something called unconventional protein secretion. The researchers found that USIC basically hitches a ride in these little membrane bubbles, or vesicles, that are marked by a protein called SYP61. It essentially sneaks out the back door in an unmarked van.
And once it's out, where does it go?
First, it ends up in the apoplast.
Okay.
That's the space between the cells. And from there, it gets into the plant's vascular system.
The plant's highway system.
And specifically into the phloem. That's the network of tubes that usually carry sugars down from the leaves to the rest of the plant.
And they proved it actually travels.
They proved it with a really cool grafting experiment, a bit like Frankenstein's monster.
Okay, paint the picture.
They took the top of a plant, the scion, that was engineered to make USIC that glows green under a special light. And they grafted it onto a normal, non-glowing rootstock.
So glowing top, normal bottom.
Exactly. Then they just waited. When they checked the roots of the normal plant later, they were glowing. The green UC had physically traveled all the way from the top of the plant down into the roots. Yeah. And when they sliced the stem, they found it concentrated right in the phloem.
Wow. So it's definitive. USIC is the messenger and the phloem is its delivery route. That explains the speed.
It does. It's much faster than passing a message from cell to cell. This brings us to a good spot for our first recap. We've covered how the signal is sent.
Good idea. So here are the key takeaways so far. First, we're talking about SSIM, systemic stomatal immunity. It's an incredibly fast lockdown of leaf pores that happens in under 3 hours. Second, the signal is a tiny peptide named USIC, which is encoded by a hidden track, or uRF, something we used to think was junk. And third, this USIC peptide gets secreted out of the cell unconventionally, jumps into the phloem, and physically travels to distant leaves.
A perfect summary. So now the messenger has arrived. It's at a distant leaf, leaf 7. It hops out of the phloem and floats over to the guard cells.
But the guard cell is a locked fortress. How does it know USIC is a friend and not a foe? It needs, what, a receptor, some kind of doorbell?
A doorbell is a good way to think of it. And to find it, the team used the synthetic USIC peptide as bait. They basically went fishing in a soup of cell proteins to see what would bite.
And what did they catch?
They caught a pair of proteins, receptor-like kinases, or RLKs. Think of them as antennas sticking out from the cell surface.
A pair of them.
A dynamic duo. The main one is called SRARK1, and its partner is CAN7.
SRK1 and CAN7, okay.
Now, normally these two just float around near each other on the cell surface. They don't really interact. But here's the key. USIC acts as a kind of molecular glue. It binds directly and very tightly to SRK1.
It grabs on.
And that binding event immediately recruits CAN7 to join the party. USIC physically pulls them together into a complex. It forces them to hold hands.
So USIC is the matchmaker. It grabs SRK1, which then grabs CAN7.
Exactly. And without that handshake, nothing happens. They made mutant plants that were missing either SERK1 or CAN7. Those plants were completely blind to the UZ signal. You could flood them with it. Their stomata stayed wide open.
And this is a really important point for you listening. Could those blind plants still defend themselves if you attack them locally?
Yes, and that is a crucial detail. If you put bacteria directly onto the leaf of a SERK1 mutant, its stomata close just fine. That's because local immunity uses a totally different receptor called FLS2.
So you have two different systems. FLS2 is for there's a threat right here on me.
Right.
And SARK1-Kina7 is for I'm getting a report of a threat somewhere else in the building, locked down as a precaution.
Precisely. One for the room you're in and one for the entire building. It's a fantastic evolutionary redundancy.
Okay, so the handshake happens on the surface of the guard cell. SRK1 and KN7 are locked together. How does that signal get from the outside of the cell to the inside to actually, close the door?
This is where things get really dramatic. Because for the signal to work, KN-theta has to move.
Move where?
It has to get from the plasma membrane, the outer skin of the cell, all the way to the tonoplast.
The tonoplast, that's the membrane around the vacuole, right? The big water sac in the middle of the cell.
Correct. The vacuole is the huge water balloon that gives the cell its structure.
Yeah.
And it's the key to how stomata work.
I vaguely remember this from high school bio. When the guard cells are full of water, they get plump and bow outwards, opening the pore. To close it, they have to lose water and go limp.
That's it exactly. To close the pore, you have to deflate the balloon. You have to dump the water out of the vacuole.
Yeah.
But KN7 is stuck on the outside of the cell. It's anchored there. You can't just walk over to the vacuole.
So how does it get free?
It gets cut.
It gets cut.
Yep. The handshake between SRK1 and KN7 triggers a massive, sudden burst of calcium ions inside the cell. It's a violent spike. And this calcium spike wakes up a dormant enzyme floating in the cytoplasm.
A molecular scissor.
A protease called MC4, metacaspase 4. This scissor is calcium dependent. It's off until that calcium spark hits it. And as soon as it does, MC4 activates, hunts down KIN7, and snips it in a very specific place.
That's amazing. Where exactly does it cut?
It's incredibly precise. It cuts KIN7 at one single amino acid, arginine 288. And they prove this by making a mutant where they swap that arginine for something else. That mutant KIN7 could never be cut by MC4.
And if it can't be cut?
It never moves to the vacuole. It's like a dog whose leash is still tied to the fence. And in those plants, the systemic stomatal closure completely fails.
Wow. So KIN7 gets sacrificed in this process. It's a one-way ticket.
It's a one-way trip. This is an emergency brake. You don't pull it unless you really have to.
So this severed piece of KIN7 is now free. It travels through the cell and arrives at the vacuole membrane, the tonoplast. What does it do there?
It goes to find the proteins that control the drain plug, specifically two of them, AHA1 and PIP21.
Okay, let's translate that.
AHA1 is a proton pump. It basically creates an electrical charge across the membrane. And PIP21 is an aquaporin. It's a dedicated water channel.
The pump and the drain.
Exactly. The cleaved KIN7 fragment gets there and interacts with them. The thinking is that it phosphorylates them, flips their switch to reverse the flow. It forces ions and water to rush out of the vacuole.
And when all that water leaves so suddenly,
the vacuole collapses. The researchers actually watched it happen. Under the microscope, they saw the vacuoles just crumple.
The balloon deflates, the guard cells go limp, and the pore slams shut.
Lockdown complete.
That is one of the most incredible biological cascades I have ever heard of. I want to pause and do a second recap because that was a lot of moving parts.
Good idea. Let's walk through that signaling chain one more time. Okay, so step 1. USIC arrives and acts as glue, binding SIRK1 and KIN7 together on the cell surface. Step 2. This handshake triggers a big spike of calcium inside the cell. Step 3. That calcium activates the molecular scissor, MC4, which cuts KN7 at a specific spot. Step 4. The severed piece of KN7 is now free and travels to the vacuole membrane. Step 5. There, it activates the pump and the water channel AHA1 and PIP21 to drain the vacuole. And finally, step 6. The vacuole collapses, the cell goes limp, and the stoma closes.
And this entire sequence, from the key in the lock to the door slamming shut, happens across the entire plant in just a couple of hours. That's stunning.
It really is.
Now, I have to ask about something you mentioned earlier. This is a close signal. Is there an open signal? I mean, biology is usually a push and a pull, a tug of war.
It absolutely is. There are other molecules like a phytocytokine called SQRU-W2 that actually do the opposite. They tell stomata to open. So the plant is constantly balancing these signals.
It's always asking itself, do I need to breathe and eat right now, or do I need to hide?
That's the calculation.
Yeah.
And they found another cool piece of evidence for specificity. We know that SRK1, the main receptor, can bind to another peptide called PIP7. But PIP7 is involved in making roots swell, not closing stomata.
Same lock, different key.
Same lock, different key, and a completely different outcome. When they added PIP7, nothing happened to the stomata. It shows that SRK1 is a really smart receptor. It's not just an on-off switch. It knows what is binding to it and sends the appropriate signal.
That's fascinating. It's all context dependent. Now let's zoom out. We've been talking about Arabidopsis, which is the lab rat of the plant world. Is this USIC peptide in, I don't know, my tomatoes, my corn?
And that is a great question. The answer is no. The specific sequence for the USIC peptide seems to have evolved only in the Brassicaceae family.
So cabbage, broccoli, canola, that family.
Right. Corn and rice don't have the gene for USIC. However, the receptors, SRK1 and kin-10-7, they're much older. They were conserved in many other plants, including the Solanaceae family, which is tomatoes, potatoes, tobacco.
Wait a minute, so you're saying tomatoes have the lock, but they don't have the key?
That was the hypothesis.
Yeah.
So the researchers did a brilliant experiment. They took the USIC peptide, the key from the cabbage family, and they just sprayed it on tomato leaves.
And what happened?
The tomato stomata closed.
No way! That's incredible. It's like finding out your car key also opens a safe in a totally different country.
It implies that SRK1 and kin-10-7 are part of a universal danger lock system. The Brassicaceae family evolved this specific USIC key, but other plants might have their own different keys that fit the same lock, keys we just haven't found yet.
The agricultural potential for this seems enormous. I mean, if you can just spray a peptide on a crop.
You could have a totally green immune booster. Instead of a harsh chemical pesticide, you spray this. The crop preemptively locks all its doors. The pathogen lands, but it can't get in. It just starves on the surface.
And it's a peptide, so it would just break down naturally. It's not a forever chemical.
It's just protein. And since it works across species, you could, in theory, use this signal derived from the cabbage family to protect a tomato crop.
You could design a master key, a peptide that works on all major crops to trigger this lockdown. Imagine a drone flying over a field, detecting the first signs of a fungal spore, and just telling the whole field to hold its breath for a few hours.
It would fundamentally change how we manage crop disease. But for me, the most profound part of this whole story isn't just the application, it's where the signal came from.
From the junk DNA.
We ignored URRFs for decades, we wrote them off as noise, and now we know they can encode these incredibly potent mobile signals that coordinate the defense of an entire organism. It makes you ask, how many other signals like USIC are sitting in the data right now just waiting for us to look?
It's like we found a hidden library, and we're just now realizing that the books contain the emergency evacuation plans for the entire building.
It completely changes the game.
So here's a final thought for you to chew on. If the cabbage family evolved this specific password USIC to use a lock that's almost universal, what's the password for corn? What's the password for wheat or for an oak tree? What other hidden tracks are sitting in the genome just waiting to be played?
The answers are definitely out there. We just have to get better at listening.
Next time you see a plant looking a little bit under the weather, remember it might not just be wilting, it might be frantically sending a telegram through its veins, telling the rest of the plant to batten down the hatches. Thanks for taking the deep dive with us.
See you next time.